So here the next part of the discussion, that is second unit of the discussion, is that is proteins, amino acids, and nucleic acids. So here the word proteins means it is of prime importance. Of living system, these are the polymers, biopolymers. Proteins are the biomolecules made of, that is, proteins are the biomolecules made of long chain alpha amino acids. Sources of proteins, these are present in cereals, pulses, fish, meat, egg, etc. These proteins, the main function of proteins is growth and maintenance of living system. Proteins upon hydrolysis, they give partial hydrolysis. Proteins gives amides on complete hydrolysis. Complete hydrolysis gives amino acids. So if you go for an amino acid, what are amino acids? If you go for amino acids are, that is, alpha hydroxy. All amino acids are a part of proteins, and they are made up of, all amino acids, they are made up of, that is, part of proteins, and they are made up of amino group. They contain one amino functional group and they contain one carboxylic functional group. The two main functional groups involved in amino acids are, that is, amino functional group and carboxylic functional group. If you go to the general formula, of amino acids. Go for the general formula of amino acids. If you take, that is, you can take R of, or else you can just start with NH2 of CH of R of COOH. So that means this amino acid, if you take, it contains one primary amine, it contains one primary amine, it contains one alkyl group, it contains one carboxylic group, and also it contains one simplest hydrogen. That means amino acid is an optically active compound. If you take an amino acid, if you consider the carbon as stereogenic carbon, then you find that it contains one carboxylic group, it contains one hydrogen, it contains one NH2 group, and also it contains one alkyl group. So 4 groups are different. We can consider the carbon as asymmetric. We can consider the carbon as asymmetric carbon. It is also taken as chiral, and we can call this as optically active compound, and finally it is going to give the racemic mixture. It gives the racemic mixture. So here if you go for amino acids, the general properties of amino acids, all amino acids, if you take all amino acids, the simplest amino, let us go like this, the simplest amino acid is glycine. The simplest amino acid is glycine, and we can simply call it as the structural formula NH2 single bond CH2 of COH. Simply we call this as alpha amino carboxylic acid. Alpha amino carboxylic acid. The word glycine means, gly means it is sweet, amino acid. Similarly, if you go for the next amino acid, that is, the amino acids are also derived from their source. If you go for, that is tyrosine means it is a source, that is cheese. Cheese is a source, that's why the word tyro represents the word cheese, and its structural formula, if you take tyrosine, structural formula, if you take, so if you go for tyrosine structural formula, if you take the benzene ring, it contains one alcohol and CH2 group, CH of NH2 of carboxylic acid. This is the structure of tyrosine. So as a matter of fact, if you go for amino acids, these amino acids, they are characterized by, because they contain both the NH2 group as well as the carboxylic group. So all we can say is all amino acids are amphoteric. Amino acids are they are amphoteric in nature. They have amphoteric nature because NH2 group is a basic site. And carboxylic group is an acidic site. Next is, if you go for amino acids, they are soluble in water. That's because they can show hydrogen bonding. Hydrogen bonding occurs between NH2 and carboxylic acid. Third is, if you go for amino acids, these amino acids, if we take, they form peptide bonds between two amino acids. Always the bond formed is called peptide bond. Peptide bond is one of the most important characteristic feature of an amino acid. So if you go for this peptide bond, the peptide bond is shown by carbonyl groups, carbon double bond oxygen with the NH and a single bond. This is called the peptide bond. So this peptide bond contains something like amido group, or it's an amide, substituted amide group. So we can find that the amino acids are always characterized by the formation of peptide bond, and if 2 amino acids are involved it is dipeptide, 3 amino acids are involved it is tripeptide, 4 amino acids are involved it is tetrapeptide. If many amino acids are involved, then we can call it as polypeptide. So whatever may be the case, this formation of peptide bond in the amino acids is one of the most important characteristic feature where we can understand in the amino acids. Now based on the occurrence of, go for the occurrence of amino acids, based on the occurrence of amino acids, these amino acids are classified into, that is, essential amino acids and which are essential amino acids are those which are not synthesized in our body, not synthesized in our body are essential amino acids. Non-essential amino acids, non-essential amino acids are those which are already synthesized in our body. So take some examples. If you go for examples of essential amino acids, if you go for examples of essential amino acids, if you take, when you go for essential amino acids, there are 10 essential amino acids and 10 non-essential amino acids. So 10 plus 10, total 20 amino acids are known to us. There are actually hundreds of different amino acids are known to us, but among those different amino acids which are known in many number, but we take only 20 amino acids. In the 20 amino acids, 10 are called essential amino acids, remaining 10 are called non-essential amino acids. And if you go for examples of essential amino acids and non-essential amino acids, let me give you some examples of essential amino acids. So if you go to the essential amino acids, what is interesting here is every amino acid is given by a 3-lettered word, 3-lettered symbol. We call it as, because amino acids are also a part of proteins, and these proteins also, they are responsible for the genetic code. So because they carry the information from one part of the body to another part of the body, and they are also involved in the various metabolic activities. Proteins are the essential amino acids, and they are responsible for giving us proper strength in our body, and because they are the energy carriers, that's why these amino acids, they are given, for convenience, they are given 3-lettered code. This 3-lettered code for a given amino acid, if it is shown, for first one, for example, LEU stands for leucine, ILE stands for isoleucine, then ARG stands for arginine, then LYS stands for lysine, then THR stands for threonine, and MET stands for methionine, PH stands for phenylalanine, then TRP stands for tryptophan, then HIS stands for histidine, then VAL stands for valine. All this comes under the essential amino acids. So these are all essential amino acids. If you go for non-essential amino acids, glycine, alanine, proline, cysteine, then tyrosine, then glutamine, then asparagine, glutamic acid, and serine are the non-essential amino acids. Among all this, if you take among all this PRO is the only amino acid. If you go for PRO, that is exceptional, PRO is proline, is the only amino acid which contains secondary amine as the functional group in its structure. What is the structure of proline? If you go for proline, it gives a genetic code PRO. If you go to the structure of the proline, you can just take it like this, that is, structure is like you can take NH of it's like this. You can take, this is the carboxylic acid, and here you have the secondary amino functional group. So this is the secondary amino functional group. Rest of all the amino acids, they have primary amine as the functional group. So here in all these essential and non-essential amino acids, one thing what we have to understand is some structures are important, some structures are not that important. Glycine, alanine, then cysteine, tyrosine, then glutamine, aspartame, then glutamic acid, serine. In fact, all the amino acids, they are equally important in their structural aspect. So we will try to write the structures of the essential amino acids and non-essential amino acids depending upon their composition, depending upon the structures. We will try to study few amino acids regarding to their structural aspect, because knowing the structures of amino acids is always an advantage, because with these structures we can find out what kind of peptide bonds are formed and what kind of the linkage it has. The amino acids are starting with is that amino acids an N-terminal amino acid or the carbon terminal amino acid. In fact, if you take every amino acid in its structure, it has to start with the N-terminal amino acid and it has to end with carbon terminal amino acid. In between the two amino acids, we can obtain the peptide linkage by the elimination of water molecules. So as we have discussed here about the aspects of essential amino acids and non-essential amino acids, if you look into the essential amino acids, out of 10 essential amino acids, I have just brought into the notice of some of the important essential amino acids, random pick. In this random pick, I have picked up valine, it has a 3-lettered code VAL, leucine, it has a 3-lettered code LEU, and isoleucine ILE, and M, that is methionine MET. So what you are noticing is, if you go for essential amino acids, if you go for valine, you are finding that here it has one primary amino group, it has one carboxylic acid, and it has one isopropyl group. You can just notice this group, we can call it as the isopropyl. It contains one isopropyl group, one amino group, and one carboxylic group, is the characteristic feature of the valine. You go for leucine. In the leucine, you are finding that there is isobutyl group. This is isobutyl. This is the isobutyl group. And of course we have one primary amine and one carboxylic acid. And in the isoleucine, this is secondary butyl. This structure we can call it as, this is secondary butyl group with the NH2 group and the carboxylic group. This comes under the essential amino acids. So likewise, we can go for the remaining essential amino acids also, and we can identify what are the important groups which are attached. See, in all the amino acids, the carboxylic group and the amino group is common. Only thing is that the third substituent, the first two substituents are same. I mean to say, in all the cases you find that amino group is in the alpha position. Whatever the amino acid you take, the amino group is in the alpha position. Whatever the amino acid you take, it is in alpha position, except for the proline. Here also it is alpha position, here also alpha position, here also alpha position. Wherever you take, the amino group is in the alpha position. That's why sometimes we call the proteins as linear condensed polymers of alpha amino acids. We call proteins as the linear condensed polymers of alpha amino acids. That's because in almost all the structures of amino acids, whether they are essential or non-essential, you find that the amino groups are always at the alpha position. So here what we are noticing here is, so similarly you go for non-essential amino acids. If you go for glycine, glycine is the simplest amino acid, wherein which you have that is the amino group is attached at the alpha position, and glycine is the only amino acid which is optically inactive, because the carbon is not the chiral carbon, it is achiral carbon, and it has two similar atoms present on the chiral carbon. That's why it can be optically inactive. If you go for the next amino acid, that is serine, again alpha amino acid, and we have a primary alcohol. This CH2OH, we call it as the primary alcohol. We call this as primary alcohol. Then in the alanine, you can see apart from the alpha amino acid, the group which is attached is an alkyl group. that is methyl group. So in this way you go for proline. If this is the alpha carbon, at the alpha carbon you find that the secondary amine is substituted. This is an exceptional amino acid, which is the only amino acid which contains secondary amine apart from the primary amine. So in this way we can classify the amino acids into essential amino acids and non-essential amino acids, and we can study their structures, and we can also remember the three-lettered code, that is genetic code of the three amino acids, because all the amino acids in the form of proteins also, because they are the energy carriers, they are involved in the metabolism and energy changes in the body. For our convenience, all the amino acids they are given three-lettered words. So here some more properties. Properties of amino acids. Some more properties of amino acids. If you take polypeptides, peptide chains with the less than 50 amino acids. Peptide chains with less than 50 amino acids. Peptide chains with less than 50 amino acids are called polypeptides. They are called polypeptides. And peptide chains with the greater than 50 amino acids, they comes under proteins. Then if the amino acid in an amino acid if greater concentration, if NH2 group concentration is greater than the carboxyl group concentration, if the concentration of amino group, NH2 is primary amine and COOH is the carboxyl group. So you know very well that this is acidic functional group and this is the basic functional group, amino group. So overall then the amino acid is going to become the basic amino acid. Basic amino acid. Examples you can take. Histamine you can take, then histamine you can take, tryptophan are the best examples of basic amines. Similarly, in an amino acid if greater concentration of the carboxyl group is present than the amino group, then you find that such amino acid is going to become overall acidic amino acid. So if it is acidic amino acid, take some examples of acidic amino acid. That is, you can take, the best example is cysteine and asparagine. Asparagine are the best examples of the amino acid which is overall acidic in nature. So in this way we can classify the amino acids, and if you find an amino acid in an amino acid if concentration of NH2, that is basic group, is equal to the concentration of carboxyl group, then such amino acid we can call it as the neutral amino acid. You can call that amino acid as neutral amino acid. The next part of the discussion of amino acids, that is amphoteric nature of amino acid. We call the amino acid as amphoteric nature because if you take the structure of amino acid, it contains one basic site and it also contains one carboxyl group with an alkyl group. This is the structure of amino acid. This is the structure of amino acid, and this amino acid contains the basic site and it also contains the acidic site. But you know very well that all amino acids, they are highly soluble in water. That's because water is a polar solvent. It is supported by the proof. Since it has high dipole moment. It has high dipole moment. Then it is insoluble in organic solvents. Then because of its highly solubility in water, this amino acid, it forms a dipolar ion. It forms a dipolar ion also called Zwitter ion. When once it forms the dipolar ion or Zwitter ion, you find that this amino acid in the form of Zwitter ion contains one conjugate base and also it contains one conjugate acid. So that means this conjugate base and conjugate acid of the amino acid during the formation of Zwitter ion is also understood in the form of isoelectric point, where the pH of the Zwitter ion of amino acid remains constant.